My name is Millard Susman, and I am a retired professor of genetics. And I have been asked to give a talk on Joshua Lederberg and the work he did here at the University of Wisconsin. He was one of our stars. He was born in 1925, died in 2008. Here's a picture of him in his lab in the genetics building, which was directly across the street and is now a biochemistry building. It started off as an agronomy building. It became a genetics building, then it became an agricultural journalism building, and now it's part of the biochemistry complex. We at the University of Wisconsin have had 4 Nobel Prizes awarded to people in the biological sciences for work that they actually did here. One of those, the first of them, was awarded to Josh Lederberg. Following that, Har Gobind Khorana and Howard Temin and Oliver Smithies. Oliver left the university before he was awarded his Nobel Prize, but all the work he did for which he won the Nobel Prize was done here. Josh just about left the university. He got the Nobel Prize in his last few months, I think, at the University of Wisconsin, and then went off to Stanford. Anyway, he is one of our stars at the university in genetics, and I thought I ought to give you some idea of where he came from and what he did here to earn his Nobel Prize. These are his parents. They came to the United States from Israel in 1924, moved into a new place in New Jersey. They were in New Jersey for a couple of years, and Josh was born there. Then they moved to New York City, where Josh grew up. Zvi Lederberg was a rabbi, Orthodox Jewish rabbi. I think he wanted Josh to be a rabbi, but Josh decided he had other things in mind, and he went on to be a Nobel Laureate in biological sciences. This is a picture of him at the age of 6 with his mother. She kept a questionnaire that Josh filled out. He was a precocious youngster at the age of 6. He wrote on his questionnaire where it said, "What do you want to be when you grow up?" He said, "I want to be a scientist like Einstein and discover a few theories in science." He was reading science books when he was in grade school. When he got to high school, he was reading biochemistry textbooks at the age of 12. He read E.B. Wilson's classic book, which was 1,200 pages long while he was in high school, on the cell. His high school was Stuyvesant High School in New York City, famous high school which has produced several Nobel laureates and a lot of outstanding scientists. At the time he went there, it was an all-male school. It's now a co-ed school, of course. He graduated at the age of 15 in 1941. While he was in high school, he started trying to do experiments, and he got interested in cytochemistry, doing study of cells by treating cells with chemicals that stain the various components of cells, and different chemicals stain different components. And people at the time, back in the 1940s, were hoping that somehow or other these stains would reveal something about the secret of the structure of the cell and how a cell worked. Nobody really understood much about how the stains worked in the first place, so they didn't reveal very much about how cells worked, but they did reveal quite a lot about how cells were put together. While he was in high school, Josh took an IQ test on which he didn't give us the score, but he scored the highest score in the Northeast region of the United States. So when he was asked, were you an exceptionally good student, he said, well, you can take that datum and decide for yourself whether I was a good student or not. I guess I was. When he left high school, he entered Columbia University. And as an undergraduate student, he talked himself into the laboratory of Francis Ryan, who was, at the time, I guess still is, a famous microbiologist. He was working on the fungus Neurospora. It's a common mold that's still commonly used in biological research. Just at the time that Josh was getting into research in Francis Ryan's lab, there was word coming from Rockefeller Institute, which is now Rockefeller University, that Oswald Avery had managed to use DNA to transfer genetic traits from one strain of pneumococcus to another. This was called transformation. And Avery showed that specific genetic traits, specifically the capsule that surrounds a normal infective pneumococcus, could be transferred from an infective pneumococcus to a non-infective pneumococcus just by treating it with DNA. So there was some specific genetic trait that was transferable with this simple molecule. At the time that Avery announced this to the world, most people in biology, I think, didn't believe it. At least there was tremendous opposition to the idea that DNA could carry genetic information, because it was considered to be a very simple molecule and it wasn't complex enough to contain all of the fancy information that must be involved in determining genetics. Josh, on the other hand, and Francis Ryan believed that DNA was a genetic material, and they decided that they'd see if they could do genetic transformation in Neurospora. So Josh's first exposure to research in the laboratory with somebody who was really a qualified researcher was to try to isolate DNA from Neurospora and use it to transfer genetic traits to other Neurosporas. That didn't work, as it happens, but at least he got some laboratory experience. And then he thought, well, it looks as if bacteria have some sort of genetic system. Avery tried to transfer some specific trait from one bacterium to another and managed to do it. Maybe he could do it with bacteria. And he decided that he'd use E. coli, which was a non-pathogenic bacterium, normal component of the human gut. At least most E. coli strains that you isolate from the human gut are non-pathogenic. So he started working with E. coli, and Ryan suggested that maybe he should go to Columbia University, where a new field of biology was beginning, biochemical genetics, in the laboratory of Ed Tatum. Ed Tatum and George Beadle in 1941 had published the first paper on the biochemical genetics of Neurospora. And Tatum was busy working on the biochemical genetics of Neurospora at Yale. And Josh took a couple of years, actually it was a couple of months, to visit the lab with Tatum at Yale and start work trying to look for sex in bacteria. And within a very short time, just a few weeks, he discovered that he could get recombination of genes in bacteria. I'll talk about that in just a minute. So he He was in fact expecting, after he got his degree at Columbia, to go to medical school. He had entered medical school at Columbia. He had stayed in Ryan's lab, took a break from Ryan's lab to go to Yale. And while he was at Yale, as I say, he discovered sex in bacteria. He was then going to go back to medical school, and Tatum said to him, you know, what you've done here in my laboratory is really PhD level work. We could maybe talk the faculty here into giving you a Yale PhD on the basis of the work you've done in my lab. And the faculty at Yale turned out to be gullible enough to say, yeah, we'll do that. You've been to all of our seminars. You seem to know everything there is to know. He was very bright and very well read. And the work you've done seems to be world-class, important research. So if you just pay tuition for the year that you've been here, retroactively, we'll give you a PhD from Yale. And so Josh got a PhD from Yale, and on the basis of at least the work that he started at Yale, he eventually received the Nobel Prize. So I'm going to talk a little bit about the discovery of sex in bacteria. What do we mean by sex? Well, for a geneticist, this drawing that I have done is evidence of sex. If you have two creatures that have different, presumably genetically determined traits, these are traits that can be inherited by their offspring, and you put them together. For example, you put them in a hen house and you leave them there for a couple of years, deliver some seed from time to time for them, and then you come back and you look at the animals that have arisen as a result of whatever goes on in the hen house, and there are now a whole bunch of chickens in the hen house, and you find some that combine traits from one parent with traits from another parent. So this guy has a head that looks like this one and has feet that look like this one. That's sex. It doesn't matter what the mechanism was by which that happened, just the ability to produce an organism that combines genetically determined traits from two different parents is sex. And that's what Josh Lederberg was looking for in bacteria. So how did he do it? Well, he isolated mutants that were that affected the nutrients that were required by the bacteria. So here he is working with E. coli. This is an E. coli cell. They look like that. They're sort of rod-shaped cells. They're capable of growing in what's called a minimal medium, which is a medium that contains only salts and a simple sugar and some trace elements that leach out of the glassware, that sort of thing. They can actually grow and produce offspring in the presence of this very simple medium. So they can produce all of the amino acids and vitamins and so forth that they need in order to be able to grow. But you can isolate mutants that require that you add nutrients to the medium in order for a bacterium to be able to grow. And Josh isolated mutants that couldn't grow on minimal medium. And in this case, this was a double mutant. This mutant has a little b here because it requires the vitamin biotin in the medium in order to be able to grow. And it has an m because it requires the amino acid methionine in order to be able to grow. But if you give a minimal medium plus just these two additives, biotin and methionine, it can grow and make offspring. So this is a double mutant that requires just two nutrients in order to be able to grow. And he isolated other mutants that required the amino acid threonine and the amino acid leucine to grow. So they wouldn't grow in minimal medium, but in minimal medium, supplemented with threonine and leucine, they could grow. He isolated double mutants like this because he knew from working with Neurospora, in fact, his first published paper was on the subject, that nutritional mutants in Neurospora were capable of back mutating so that they didn't need nutrients anymore, that they were now able to grow in minimal medium again. And that happens about out of one cell out of a million, that the mutant can revert to wild type. And since Josh wasn't sure how much sex bacteria were capable of, how many offspring they could produce that were going to combine genes from two parents, he put together double mutants, figuring that if this mutation reverted to wild type one time out of a million, and this one reverted to wild type 1 out of a million, and if those things happen independently of one another, then it would be 1 out of 10 to the 12 that you'd get both of them reverting at the same time. That's more bacteria than you usually use in an experiment, a whole lot more. So he thought, well, if I can get one recombinant out of 10 to the 6 bacteria, and the background for double mutation is 1 out of 12, that's good enough to detect recombination. So that's what he did. He mixed together a biotin methionine requiring bacterium with a threonine leucine bacterium, put them in a medium that was supplemented with everything that was needed by both of these bacteria so they could grow into a big mixed culture. Most of the cells in that culture he expected would be either little b, little m, or little t, little l bacteria. But some of them, it turned out, when he plated them on minimal medium, were capable of growing on minimal medium. So his mixture of cells produced about 1 out of a million that had the genes from the two parents that made it possible for the bacterium to make its own biotin, its own methionine, its own threonine, and its own leucine. So that was the first demonstration in Tatum's lab that bacteria were actually capable of some sort of sexual process that produced offspring that combined genetic traits from both. He was immediately famous as a result of this experiment. And the word got out. He was thinking of going back to medical school, but the word got out that there was this terrific, bright guy who was capable of crossing bacteria and making genetic recombinants, and who intended to devote his career to studying the genetics of bacteria. And here at Wisconsin, the news somehow electrified the faculty in the genetics department, and they said, we've got to get this guy. Actually, they argued for quite a while about whether they wanted to get this guy or not, because he was a New York Jew who had never been on a farm, and this is a college of agriculture. And so there was some discussion about whether you wanted this strange misfit to work in a laboratory where we were interested in corn and mink and cows, and here was this guy working on bacteria who had never seen a farm in his life. However, once they met him, they realized that he was really a catch, and they went for him and hired him as an assistant professor. So instead of going to medical school, he became an assistant professor at the University of Wisconsin. So what did he do while he was here? One of the first things he did was discover not only that there is sex in bacteria, that is, that they're capable of producing offspring that combine genes from two different bacteria, but that there are sexes in bacteria. And he discovered that in conjunction with his wife, Esther Lederberg, and with this guy, Luca Cavalli-Sforza, who was a postdoc in the laboratory. This is a picture of them at the Peninsula State Park in Door County, enjoying a little break from the laboratory. This is what they discovered, that if they tested a lot of different bacteria, independently isolated mutants of E. coli in their laboratory, that they could divide them into two kinds of bacteria. There were F plus bacteria, which were called, that's fertility plus, and F minus bacterium that were fertility minus. So if they put appropriate mutant genes in these bacteria so that they could detect recombination, they could detect the production of offspring that had genes from two different parents. When you crossed an F minus with an F minus, they didn't produce any recombinants at all. If you crossed an F plus with an F minus, you got recombinant offspring. And if you crossed an F plus to an F plus, you got recombinant offspring. So there were bacteria that could initiate what was then called conjugation, and bacteria that couldn't, and they were the F minus bacteria. And then they made all kinds of discoveries about bacteria. This is bacterial conjugation. Josh did a lot of work to demonstrate that, in fact, the production of recombinants in E. coli and the system that he used required the bacteria to somehow come into contact with one another. You couldn't take the medium from an F plus bacterium and put an F minus bacterium into it and have it pick up genes from the F plus. You actually had to have the bacteria touching one another in order to be able to produce recombinant offspring. And it turned out that when you had an F plus bacterium, it was hairy. These projections that come off of this bacterium are called pili. Singular is pilus. It's Latin for hair. So you have hairy male bacteria. These turns out, are what are called male bacteria, and hairless female bacteria. This is an F minus cell. And the pili form connections between the F plus and F minus cells, and then the cells get together and form a conjugation bridge through which genes are passed from a donor F plus bacterium to a recipient F minus bacterium. What they discovered first was that when you mix F plus and F minus bacteria with one another and then separate them, the F minus bacteria become F plus. So the ability to serve as a genetic donor in an experiment or an initiator of conjugation is infective. The F plus bacterium can infect an F minus bacterium with the ability to become an F plus, to initiate conjugation. And it turned out after much work that the explanation for this was that there was in an F plus bacterium, this is a sort of diagrammatic picture of a bacterium, and this circle of DNA is the bacterial chromosome, and this circle of red DNA is called an F factor. And it turns out that the F factor, and the thing that makes this an F plus bacterium and that gives it the ability to grow pili on its surface and to transfer genes to another bacterium, though the ability to do that is all contained in the genes on the F factor, which is separate from the chromosome and which confers these new genetic properties on a bacterium. When you put the two bacteria together, an F plus cell with an F minus cell, a conjugation bridge is formed, and the F factor is replicated, and a copy of it gets transferred to the F minus cell. And then the F minus cell becomes an F plus cell. This was a sort of astonishing discovery that sex in bacteria was determined by something quite different from the sort of thing that determines sex in higher organisms. When Josh started all of this, he thought, these things are going to act like eggs and sperm. You know, you're going to get two bacteria that come together and they form a zygote somehow, that they fuse and they go through meiosis and they do all of the things that higher organisms do to produce offspring. But in fact, It's much simpler than that. It's just that two cells get together and one cell transfers from some DNA to the other cell, and then the other cell incorporates that DNA. In the case of F factor, it just incorporates F factor. However, it was also discovered that the F factor can interact with the bacterial chromosome. So this is a bacterial chromosome, DNA. Bacteria have circular DNA chromosomes. And here is the F factor. It can interact with the chromosome and get incorporated into the chromosome as part of the chromosome. So this has a black chromosome from the bacterium incorporating a red little chromosome, mini chromosome from the F factor. This thing is now what's called an HFR cell. And these cells were discovered independently by Joshua Lederberg and Cavalli Sforza here at Wisconsin, and by Bill Hayes and his collaborators in London. So there were two kinds of HFR cells that were worked on for some years. One of them called HFRC, which was isolated here. That stood for Cavalli. And one of them was HFRH, and it was isolated in London, and it was, the H stood for Hayes. Well, what happens when you mix one of these guys, an HFR cell with an F- cell, is that it initiates transfer of DNA, but since the F factor is incorporated into the chromosome of the bacterium, it starts off transferring a little bit of copy of the F factor, and then it goes on and copies around the chromosome, and it copies DNA from the bacterial chromosome as well. So a little bit of F factor gets transferred to the female cell, and then a whole bunch of chromosomal DNA, and then a little bit more of the F factor at the end. Bacteria like this are called HFR because they give a high frequency of recombination, HFR. And in bacteria like this, genes that are close to where the F factor is incorporated into the chromosome get transferred to the recipient cell at almost 100% frequency, whereas ones that are farther down the chromosome don't. It takes about 90 minutes for this process to take place, for the whole chromosome to be copied and transferred to an F- cell. And these cells can separate at any time during that mating process, so that generally only a little piece of bacterial chromosome gets from the donor to the recipient cell, but some of the chromosome gets to almost all of the cells, all of the F- cells that are stuck to an HFR. And then once the piece of DNA is transferred to the recipient chromosome, it can interact with the recipient chromosome, and the bacterial DNA from the donor can get incorporated into the chromosome of the recipient so that you produce a genetic recombinant. All of that stuff was explained and discovered by Lederberg and his colleagues here at Wisconsin. Finally, Lederberg did give up on the idea that these two cells fused and had a meiosis and went through the kind of recombination that higher organisms do, and accepted the idea that in fact there was one cell that transferred DNA to another cell in order to produce recombinants. Okay, so that was one set of experiments that Lederberg and his collaborators did here. They discovered how sex works in bacteria. They discovered the existence of these little extra chromosomal pieces that are called F factor. And in general, it turns out there are a lot of those things, a lot of F factors in bacteria in nature, or a lot of little extra rings of DNA. And that they're not all sex factors. Some of them can initiate transfer to another cell, and some of them can't. Some of them carry genes for just making hair and mobilizing the bacterial chromosome. Some of them contain genes for antibiotic resistance. So there are a lot of these things like that in natural bacteria. And in general, these little extra pieces of DNA that can replicate separate from the chromosome inside a bacterial cell and that are inherited by all of the progeny of that bacterial cell are called plasmids, which is a word that was made up by Josh Lederberg. Now here's another, I'm going to change subjects entirely now. I want to talk about another kind of experiment that Josh did here with colleagues. Question, how do mutations arise? If you expose bacteria to antibiotics, you get a very small number of antibiotic-resistant bacteria from a large population of antibiotic-sensitive bacteria. How did those mutations get there? Were they already there when you added the antibiotic? Is that what made it possible for some cells to survive and grow in the presence of antibiotic? Or did the antibiotic induce the bacteria to become antibiotic resistant? Well, Josh and Esther, his wife, invented a technique called replica plating, which has become a sort of standard technique for microbiologists, to study that question, and here's how it worked. So if you spread 100 bacteria on the surface of a petri dish that contains a nutrient medium, it's got a gel, it's got chicken broth in it or something like that, you get about 100 colonies. Each bacterium makes a colony. If you put antibiotics in the agar, that's what's happened here, and you spread 100 bacteria on the surface, you won't get anything. But if you spread about 100 million bacteria on the surface of the plate, you'll get some colonies growing, and these are antibiotic-resistant colonies. All of the cells that they produce, if you take those antibiotic-resistant cells and grow them in the absence of antibiotic, they'll all be antibiotic-resistant when you expose them to antibiotics. So they are inheriting antibiotic resistance, but where did the mutation come from in the first place? Did it come from a treatment with antibiotic, or the mutation's already there? Well, this is how Josh and Esther tested that. They used a technique called replica plating, and here's what they did. They started off with a culture of bacteria, and they took a drop of bacteria and spread it on the surface of a petri plate, spread it around with a sterile spreader, and then after incubation, the bacteria form colonies on the surface of the plate. And then they used this technique, which is called replica plating, to transfer bacteria from this plate that has bacteria on it. This is a plate with no antibiotic in it. And they have a wooden block, round wooden block, with a piece of velvet attached to it. This was Esther's idea. Velvet was kind of like a zillion little needles that stick down into the surface of the petri dish and pick up a sample of the bacteria that are growing on the surface of the agar. And then you could stamp that velvet onto several other plates, take a sample of bacteria from the master plate and deposit it on a series of other plates. And if there are antibiotic-resistant cells on this plate and you transfer them to the other plates, they should be able to grow colonies on the other plates. So here's the way they did it. They started off with a master plate that was just coated with bacteria. They plated maybe 1 million bacteria and made 1 million colonies on this plate. And then they transferred a sample of that on a velvet pad to other plates that contained antibiotic. And sure enough, they would get colonies growing on the antibiotic-containing plates. The question was, were these growing from mutants that were already present on the master plate? Or were there just a few lucky bacteria that managed to become antibiotic resistant under the influence of the antibiotic on the replica plate? Well, when they looked at the positions of the colonies growing on the replica plates, they noticed that there were some, like these two green guys, that grew in exactly the same position on 2 different plates. These blue guys grew on 3 different plates in exactly the same position. These little purple guys grew on 3 different plates in exactly the same position. So that suggests that maybe there were places on this plate where there were little families of resistant bacteria that had grown during the formation of the lawn of bacterial cells on the master plate. So they could go back to the master plate and mark these places that had given rise to colonies on several different antibiotic-containing plates and pick cells out of those. They could, if there's 1 cell out of 10 to the 7 or 10 to the 8 on the whole plate that's antibiotic resistant, if those mutants were already present before the cells were transferred to replica plates, then in these places where there were cells that appeared on several different replica plates, maybe there's 1 out of 10 to the 6 or 1 out of 10 to the 5. They're little families of mutants that grew up while the cells were growing on the master plate. So Josh and Esther picked cells out of these green and red and purple marked places and spread them on new plates and did that whole process over again, making smaller and smaller inocula each time, hoping to increase the frequency of mutants among the normal antibiotic-resistant cells. And by doing that, they actually managed to isolate pure cultures of antibiotic-resistant bacteria that had never been exposed to antibiotics. And they concluded that, in fact, antibiotic-resistant mutants occur in the untreated cells. They occur spontaneously, that they're not induced by the antibiotics. And they developed a method later for doing this in a more quantitative, statistical way to show that, in fact, that was exactly right, that you could account for all of the resistant cells by assuming that there were mutations occurring spontaneously in the sensitive culture of bacteria and that they were ready to be antibiotic resistant when they were transferred to the antibiotic cell plates. Okay, 2 more discoveries from the Lederberg lab that I'll talk about pretty fast. The discovery of transduction. There was a graduate student, Norton Zinder, who worked in Josh's lab here at the University of Wisconsin. In fact, he came to the University of Wisconsin from Columbia at the same time that Josh did and became Josh's graduate student. And Josh suggested that he try to see if he could get sexual transfer of genes in Salmonella. Because if you're going to work with a pathogen, you don't want to work with it yourself. You want to have a graduate student work with it. And Zinder tried that, and he managed to get gene transfer in Salmonella, but he discovered that that didn't require cell-to-cell contact, that he could actually take the medium from one strain of Salmonella and treat another strain of Salmonella with it and transfer genes in the medium without having the cells touch one another. And on inspection, it turned out that the carrier of those genes were viruses that were growing in the first strain and that were carrying bacterial genes to the second strain of bacteria. I don't know how many of you know what a bacterial virus looks like, but here's a bacterial virus that's near and dear to my heart because this is what I worked on as a researcher. And this is bacteriophage T4, just a little tiny thing. but it has quite a complex structure. All of the things that you see here are made of protein, and inside the head is a long molecule of DNA that contains the genes that are required for making all of this stuff. And the life cycle of the virus looks like this. The virus attaches to the surface of a bacterium. It injects its head full of DNA into the bacterium through its tail. Some viruses do different things, but this is a typical virus. The DNA that's injected by the phage takes over all of the biosynthetic apparatus of the bacterium, breaks up the bacterial DNA and breaks it down to its smallest subunits, just nucleotides. And then it starts making bacteria virus DNA. And the bacterial virus DNA accumulates inside the cell. The genes that are on that DNA take over the biosynthetic apparatus of the cell to make the proteins that are needed to package the DNA, and the DNA gets packaged inside of heads, just like the one in the infecting phage. And then eventually the cell pops open, and out come 100 or so viruses. And all of that takes about 40 minutes. What Norton Zinder discovered was that in Salmonella, the Salmonella strains he used that were transferring their genes to other cells carried a bacterial virus in them, a virus called P22, which it was incorporated into the bacterial chromosome just the way F factor incorporates into an HFR chromosome. And sometimes those bacteriophages popped out and popped the cell and released a crop of offspring. And those viruses were capable, while they were replicating, sometimes of picking up a chunk of bacterial DNA instead of a chunk of virus DNA. So transducing phages of this P22 type are capable of infecting a cell or popping out of the chromosome of a cell, and as they replicate, they can take some broken pieces of bacterial chromosome and pack them into heads. Most of the viruses will contain viral DNA, but a very small number contain bacterial DNA, which they can then deliver infectively to another cell, and that DNA can be incorporated into the chromosome. So this was a whole new way of doing sex in bacteria, by doing it using viruses instead of using cell-to-cell contact of bacteria. And then Esther, Josh's wife at the time, I should say that Esther and Josh didn't stay married for very long. I think it was about 10 years, and then they broke up. And Esther, in her later years, was very angry that she didn't get more credit for the work that had been done in the laboratory than she did get. And if you look at her website, you'll find that she's devoted pages and pages and pages and pages to documentation of the contributions that she's made to microbial genetics. I don't think anybody questions Esther's contributions to microbial genetics. But unfortunately, this was, I guess, not a good relationship, and when it broke up, it left bad feelings. But one of the things that is absolutely undisputed is that Esther discovered a bacteriophage, a bacterial virus called Lambda. And Lambda became one of the most widely used viruses in microbial genetics laboratories and molecular genetics worldwide. So you can certainly give her credit for having discovered this guy. This is a bacteriophage Lambda. And it turned out that the very bacterium, the E. coli bacteria that they were using in the laboratory, contained a virus that was incorporated into its chromosome. And if you UV irradiated these bacteria, the virus would pop out of the chromosome and start replicating inside the cell and pop it open and release 100 viruses. And that it would then go on and infect other cells, but that sometimes when this virus popped out of the chromosome, it would pick up one of the genes on either side of it, either a GAL gene on one side of its attachment site or a bio gene from the other side, and transfer it along with virus DNA into a recipient cell, and it could get incorporated into the chromosome in the recipient cell. So the virus that Zinder discovered in Salmonella could transfer essentially any gene of the bacterium to a recipient cell. The virus that Esther Lederberg discovered could transfer only either the GAL gene or the bio gene, depending on how it broke out of the bacterial chromosome, and carry that gene to a recipient cell. So they discovered two kinds of sex in bacteria that involve viruses, and then the F factor induced sex as well. That's really what they did while they were working here at the University of Wisconsin. These experiments gave rise to the entire field of bacterial genetics, which was largely the area in which molecular genetics as we know it today developed. So this was fundamental work that affected research in genetics worldwide. In 1958, these three people, George Beadle, Ed Tatum, and Josh Lederberg, were awarded the Nobel Prize for having developed sex and genetic methods for working with microorganisms. These two this one for Neurospora mold, this one for bacteria, this one for working with both of these people. In 1967, the University of Wisconsin honored Lederberg by giving him an honorary degree here, and this is a picture of him with Jim Crow, the chair of the genetics department at the time, and Fred Harvey Harrington, who was the president of the university at the time, awarding Josh his honorary degree, and Josh is giving the Nobel medal that he received to the university as a gift because he was grateful to us for having made his research possible. This is his medal. We have since lost it. If it's at the university, we don't know where it is. But anyway, it's not in the genetics department, doesn't seem to be in the library, doesn't seem to be in archives, doesn't seem to be in the president's office. So we don't know. But Josh isn't here anymore, so maybe he won't hold it against us. Among other things, Joshua Lederberg organized the Department of Medical Genetics here before he left and served as chair of the department for 2 years. He greatly stimulated research in the genetics department itself, as well as the medical genetics department, so that a whole lot more molecular genetics grew up here as a result of his influence. He moved to Stanford University in 1958, right after receiving the Nobel Prize. He stayed there until 1970, and then he moved to Rockefeller University in New York as president of Rockefeller University and served there until 1990. He did a lot of other things in addition to his work in genetics. He was one of the first people to realize that biological warfare was a danger to humanity, and he rallied support from the scientific community to do something about the possibility of biological warfare and to try to get international agreements to ban biological warfare. He became an advisor to our government. He participated as a diplomat in international negotiations on biological warfare. He also got interested in exobiology, that is, the search for life on other planets. He was the first person to realize that you shouldn't send a rocket to Mars, for example, without sterilizing the rocket, because you might find Earth organisms that you've planted on Mars on your rocket. So he emphasized that it was necessary to worry about not contaminating the rest of the solar system with our gadgets. And he was a leader in that effort. And then he was a leader in trying to figure out how to analyze the chemistry of other soils on other planets to see if there was any evidence that there were living things on those planets. And among other things, he and a bunch of people who were actually computer scientists started working on a kind of primitive artificial intelligence system for analyzing spectrometry data from mass spectrometers that were sent to other planets. And Josh was one of the authors of this primitive system, Dendral, for analyzing mass spectrometry data and trying to figure out what sorts of organic molecules or space probes could find on other planets. This was actually one of the first ventures in what was then called artificial intelligence. I think it's not anymore, but anyway, he was working with the pioneers in the field of artificial intelligence. And for 5 years, he wrote an article, a column in the Washington Post called Science and Man, which was devoted to trying to educate the public about science and how it affected our lives. And it would be nice to have him back, because the support of science in the public is becoming increasingly scarce. And Josh was the sort of person, he wrote very good, interesting, and convincing columns. He was the sort of person who could have drummed up public support for scientific enterprise. So I'll just close with one more remark. I think I'm used up at least all of my time. Next week I'm going to talk about Alex Brink, who was a corn geneticist in our department and who did other groundbreaking work in corn genetics. And in his brief autobiography, he mentions that Josh Lederberg, he thinks, was his greatest accomplishment as chair of the Department of Genetics for 18 years, that recruiting Josh Lederberg to this laboratory did more for genetics here than anything else that he did during his 18 years serving as chair. So I leave you with this rather friendly picture of Josh, and I'll talk to you next week about Alex Brink. Yeah, I'd be happy to answer questions. Saturated. Tatum was a Wisconsin graduate, right.
So this isn't a question, this is a comment. You didn't say, tell this anecdote, and maybe it's not even true, but it's something that I've heard several times and think is very amusing. When his mother got the results back from his intelligence test in New York, she said, Josh, you're the smartest guy in New York. And he says, Natch.
Yes. You're right. Did you all hear that? That was a nice story. That when he got his IQ score, his mother said to him, Josh, you're the smartest kid. And it was actually the northeast region of the country. And Josh said, Natch. He wasn't a modest man. He and Bill Hayes had a disagreement about whether bacteria, when they recombined in conjugation, fused with one another and formed a zygote, like a real higher organism zygote, or whether, in fact, one bacterium transferred a little bit of DNA to a recipient bacterium. And Hayes had insisted that it was a unidirectional transfer and that these cells separated from one another after mating, and that there was no fusion of the cells and there was no transfer from the F- bacterium to the F+. And Hayes visited here and had lunch with Josh and with our friend Jim Crow, who was one of our prime geneticists in the genetics department. And they argued about this for the entire lunchtime. Bill Hayes presenting his data in his very English, very proper, very modest way, and Josh presenting his evidence in his very New York, very aggressive way. Jim said that after listening to this discussion, he was quite sure that Josh was right and that Hayes was just confused. And it, of course, turned out that Hayes was right and Josh was confused. But it took Josh several years to admit that he was wrong and that Bill Hayes had the science right. So he was a very persuasive fellow. Yeah.
Well, you mentioned Briggs' pride in bringing Josh here. Can you tell the story that Joshua was actually responsible for bringing Jim Crow here? Just mentioned.
Yeah, that's right. Jim Crow was at a meeting in Cold Spring Harbor when Josh presented his initial data on the genetic map of E. coli. And Jim was a brilliant geneticist, and he went and asked Josh some questions after the 3-hour talk that Josh gave at Cold Spring Harbor. This became a kind of legend among geneticists that there was this guy just out of graduate school who wasn't on the program, but he had an evening session that he announced, and then he got all these people, distinguished scientists, together in a room and harangued them for 3 hours about the work that he'd done. But Jim asked some questions that impressed Josh so much that he went back to Wisconsin and said, we got to get this guy here. And so he initiated a recruitment that ended up getting us not only Josh, but also Jim Crow. So it was a kind of a genetic event in itself.
Thanks very much.